If I were to be diagnosed with glioblastoma today, I will be preparing for the end of my life because the treatments available are proven not to save my life. The natural question for anyone in such a position is to ask what experimental options are available to them. Typically, experimental options only become available to me once my cancer has failed the best available treatment. In the light of this knowledge, I ask the question, why must I wait for the treatment that is known to fail before I have access to experimental treatments? The history of cancer teaches us that when patients and doctors work together, often great strides forward can be made. CUSP-ND is an international collaboration of doctors, patients, survivors, researchers, and bereaved relatives, united by a determination to bring more meaningful treatment options to glioblastoma patients today. The group came together through the process of a documentary film that charted the progress and stories of 3 very unusual long-term survivors who employed a radical cocktail strategy. A scientific investigation into the outcomes of these unusual long-term survivors appeared overdue.
When I was first contacted to discuss a cocktail approach for newly diagnosed patients with glioblastoma, I was feeling enthusiastic about this approach because we already had employed the CUSP-9 approach to patients with recurrent disease on a compassionate use basis. It made a lot of sense to me to extend this concept to patients that were newly diagnosed with this disease. The standard of care for patients with glioblastoma is palliative from the start. And that means that to make a change, a new concept, possibly or definitely new treatments are needed. One of the hallmarks of cancer in general, and glioblastoma in particular, is the development of tumor cell resistance, which is far from being adequately addressed by single-agent treatments.
This is a tumor. In its early stages, it is treatment naive and often responsive to initial therapy. As the tumor grows, it frequently acquires mutations, which make it more complicated to address therapeutically. Treatment with a single agent will often deliver an initially positive response, eliminating a significant population of the tumor cells. The problem lies in the cells that survive the initial treatment. They are reproducing rapidly, and each new generation develops improved survival mechanisms that allow these cells to resist further treatment with the same agent. The very process of sequential treatment, whilst delivering short-term benefit, may actually be counterproductive in the long term, as the tumor cells can become more complex and resistant as an unintended consequence of single-agent therapy. Although the single agents are potentially effective treatments, sequential therapy does not allow for any secondary agent to take advantage of the window of opportunity created by the primary agent. With each round of successive treatment, the surviving cell populations are increasingly well equipped to survive. Therapy becomes more difficult for the treating physician, and survival a distant prospect for the patient.
The concept of using CUSP-ND is to use readily available drugs that are used off-label and have known effects against certain signaling pathways that are operative in glioblastoma. The use of cocktail therapy makes sense because a multitude of agents is able to address potential resistance more effectively than single agents applied in sequence.
Whilst probably far from complete, scientific investigation has revealed a multitude of growth signaling pathways that are important for the survival of glioblastoma cells. However, when a treatment successfully blocks one of these mechanisms, the tumor simply shifts to other parallel or compensating pathways that have not been blocked. We term this the Nile distributary problem, in that if one of the distributary at the delta is blocked, the total flow of water into the sea remains unaltered. It may be possible to disable the survival mechanisms that the tumour cells will call upon when they find themselves under the stress of an effective single agent. By preemptively blocking the tumour's survival mechanisms, our strategy seeks to render the existing treatment more effective. We raise the question whether the efficacy of temozolomide, the current standard of care chemotherapy in use for glioblastoma, could be magnified by the correct supporting cast of drugs. Clearly, combining many drugs together comes with significant risk, but it's because of that risk that we need a clinical study, that we need a clinical trial operated by professionals to investigate whether or not this is a feasible line of treatment for patients who are so desperately in need. The long-term survivors who inspired and formed part of our research group did not discard standard of care. They couldn't afford to. Similarly, we don't expect patients today to forego standard of care. What we're trying to build is an assembly of treatments which are tolerable to those patients, to add and to improve to the existing therapy, which, whilst is the best proven available, is not saving lives.